Very good evening, everyone. Please confirm if you can hear me. Is the screen visible? Please confirm. So in the chat, you can confirm me. Good evening, Ramdas. I can see Samora joining. Thank you for confirming, Nayanika. Very good evening. Good evening, Surendra. Thank you for confirming. Good evening, Harshita, Tonio, Achal. So I hope you all are doing great. Practicing questions. So today we'll be starting with discussion of questions from Unit 13, that is Methods in Biology. And particularly in today's session of Brahmastra Series, we will be discussing the questions based on topic from subunit 13B, that is Histochemical and Immunotechniques. So I'm sure you know 13B, subunit 13B is all about immunological techniques where you study about the techniques like ELISA, radioimmunoassay, Western blotting, flow cytometry or fluorescence activated cell sorting. Then you also study about fission gish, that is in situ hybridization, immunohistochemistry, immunoprecipitation. So many techniques are there. Now, although if you go through the previous year question papers, there are certain techniques on which frequently questions are asked, like Western blotting, fluorescence activated cell sorting, or flow cytometry. But other than these techniques, the questions can be framed on ELISA also, radioimmunoassay also. So if not direct question or experimental question on a particular technique, question can be framed by combining different techniques. Like in the options, you can get ELISA also, you can get a radioimmunoassay also, along with other techniques like spectroscopy or microscopy. So the point is, whenever you are studying subunit 13B, you have to cover all the topics. Okay. So today I will be discussing questions from the topics based on the immunological techniques, and during this session you will get an idea about what are different types of questions which are asked from these particular subunit. What is the strategy of solving such questions? Is there any trick we can follow while solving such questions? Okay, so let's get started. So let's start with the question, which is you can say a part B question, the type of question which you get in CSIR net exam, a part B question. So go through the question on your screen and you can answer me. Yeah, very good evening, Hemlata. Good evening, Sunshine. I can see some more have joined. So very good evening to everybody and welcome to today's session of Brahma series. So this is the first question which I have shared. So the question is on your screen. Please go through it and tell me what could be the answer. So I request everyone to kindly solve, at least try. So one thing is very much clear: to answer this question, you need to be familiar with the techniques, like what is immunoelectrophoresis, what is rocket electrophoresis, what is immunofluorescence, what is passive agglutination, right? So once you are familiar with these techniques, at least like then you can answer this question. Okay, so majority says immunofluorescence. Okay. So basically, okay, so everyone is saying option 3. So are you familiar with the terms precipitation reaction or agglutination reaction? And what is the meaning of sensitive? Like when you say a particular technique is sensitive, like there are two terms, sensitivity and specificity, right? So whether a particular technique is effective or not, it depends upon the sensitivity and specificity, right? So sensitivity is that is the low quantity of an analyte that a technique can detect. Like when you say radioimmunoassay is a sensitive technique because it can detect as low as picogram of an analyte, right? So similarly here, to answer this question, you all need to be familiar with the two terms agglutination reaction and precipitation reaction. Now, the agglutination reaction is what? When you are mixing, like in this figure, you can see that when you are adding, like, immunoassay is what? Immunoassay is where you are using an antibody. Okay, immuno, it refers to the antibody which you are using to detect a particular antigen. So in agglutination reaction, basically it refers to the clumping of the particles together. So keyword here is clumping. So basically when antigen and antibody interactions happen, so it results into the clumping of antigen and antibody, as you can see over here. So when you are mixing an antigen with the corresponding antibody at suitable pH and temperature, and if clumping happens, we call it as a agglutination reaction. However, another kind of reaction is a precipitation reaction. So here, when you are mixing again antigen and antibody, so like at a suitable temperature and pH, an insoluble visible complex is formed. So here the keyword is insoluble visible complex. So a precipitate is formed, right? We call it as a precipitate. And as you can see here, that if your antibody is in excess, precipitation will not occur. Antibody is more than antigen. Similarly, if your antigen is more than antibody, again precipitation will not happen. For precipitation to happen, you should have the equal amount of antibody and the antigen. So here you can see this is the equivalent zone where you have equal amount of antigen and antibody and resulting into the precipitation reaction. Now in the question, 4 techniques have been given to you. First technique is passive agglutination. So passive agglutination is a technique where, so it's an agglutination, like from the name only you can make out here agglutination reaction will happen, but it is passive agglutination. So passive here, it means that your antigen is bound to a inert, any inert or a carrier molecule. It could be a any kind of matrix you can say, right? So an antigen bound to an inert or a carrier before it is made to react with a particular antibody. So here you can see this is a carrier molecule and this is the soluble antigen, right? And then finally when you are adding the antibodies, it results into formation of a clumping. So it's a passive agglutination. And so like here inert or carrier can be a latex bead. It can be a latex bead. Then another technique given was rocket electrophoresis. So as shown over here in rocket electrophoresis, we use antigens, like you add the antigens in the gel containing the antibody and you perform the electrophoresis. So basically what happens, the antigen antibody, like as the antigen it migrates, what will happen? So precipitation will happen. So here you can see these are the precipitation arcs and the shape is of rocket. So it's a quantitative technique because the height of this rocket shape, which is shown here, is proportional to concentration of the antigen in the well. So like here you can see this is the, you can find out how much is the concentration by seeing the height of this rocket. So rocket electrophoresis is a precipitation reaction. And then another technique given was immunoelectrophoresis. Now in immunoelectrophoresis, you have a mixture of antigen and this mixture of antigen you are going to separate. So first you will be performing electrophoresis to separate the mixture of components by charge. And then you will be like troughs are then cut into the agarose gel, which are parallel to the direction of the electric field. And you will be adding the antibody or the antiserum is added to the trough. And then you can see that antigen and antibody, they will diffuse towards each other and they produce a line of precipitation where they meet in appropriate proportions. So this technique is called as immunoelectrophoresis. So again, it's a precipitation reaction. And lastly, you have immunofluorescence, where we use antibody to detect a particular antigen, but that antibody is conjugated with a fluorescent molecule, a fluorochrome. What is a fluorescent molecule or fluorochrome? Which when absorb a light of particular wavelength, always emit light of longer wavelength. And this phenomena we call it as fluorescence. So here we are conjugating the antibody with the fluorescent molecule which absorbs light of one wavelength and emit light of another wavelength. Now we have to find out of these four which is the most sensitive immunoassay. So if you go through Cube, the reference book Cube of immunology, this particular table is given and your answer lies in this table. So if you see the sensitivity for all techniques, the sensitivity is given. Precipitation, it is mentioned for precipitation techniques based on precipitation reaction, agglutination reactions, and also sensitivity of radioimmunoassay, ELISA, right? Immunofluorescence also it is mentioned. So you can see if you compare precipitation and agglutination, the sensitivity is 20 to 200, right? But for agglutination, it is like zero point 0.3 if it is direct, and if it is indirect, like as I told you, indirect or like here it is inhibition, so 0.006 to 0.06. So comparatively, agglutination reaction you can say is more sensitive than the precipitation reaction. And most of you have mentioned immunofluorescence, so you can see the sensitivity of immunofluorescence. So now tell me what is the answer. What do you think is the most sensitive technique? Agglutination is more sensitive than precipitation reaction, which are the techniques like immunoelectrophoresis, rocket electrophoresis are based on precipitation reaction. And then you have the immunofluorescence. So please tell me now what you say, what is the answer here after going through this table. Yeah, very good evening, Jaya. Good evening, Yusra. Hemlata, yes. Sensitive means it can trace small amount of the sample. Yes, please answer in chat. 4 options are there, right? Yes, Achal, very good. It will be option 4. So that means your option 4 is the passive agglutination, right? Which is the most sensitive technique. Absolutely right, Surendra. Very good. See? So now you all are giving correct answer. Yes, Priyanka, very good. Sunshine, yes, passive agglutination. Ramdas, very good. So that is the importance of referring the books. Like even during our classes, regular classes, we show you these tables, right? Or even we mention you like which topic is important and which particular table or figure you should refer from the reference books, right? So make sure while revising these topics, you pay attention to these details, these tables, some figures which we always tell you during the class lectures. Sajane, very good evening. So I have just started only first one question I have discussed so far. Now moving over to another question. So you can see although the question which I discussed in the previous slide, it was part B question. It was very direct question. Only you need to memorize is the sensitivity, and that you can do by referring to one table, right? So even if you don't know about these techniques, if you have studied about what is agglutination reaction, precipitation reaction, and sensitivity of immunofluorescence, ELISA, RIA, you can answer the questions. Then moving over to another question. The question is about monoclonal antibody production by hybridoma technology. And myelinoma cells use lack the enzyme HGPRT, that is hypoxanthine guanine phosphoribosyltransferase transferase, such that few cells can only survive when selected on hypoxanthine aminopterin thymidine, that is HAT medium. So what is the role of aminopterin in this medium? So basically this particular question is based on hybridoma technology, and you need to be familiar with what is hybridoma technology, how you can develop monoclonal antibodies, and how the head selection is done. And even if you are only like entire the process is not required, like you here the question is not about how monoclonal antibodies are formed. It is already given. They have only asked you what is the role of aminopterin in the medium. So what is the answer here? 4 options are given that aminopterin can be used as cell cycle inhibitor of myelinoma cells or to block the pathway for nucleotide synthesis. Third option is to facilitate fusion of myeloid B cells and antibody producing B cell. And fourth is to facilitate production of antibody producing B cells. Please answer the question. I can see so many are attending, but only few are answering. I really appreciate everybody, those who are seeing this, those who are attending this session. please try. I'm here to help you all, right? Biotechnica has specially come up with this session, this Brahma series, basically to help you all, right? Because we get a lot of questions that regarding the question discussion, and many of you, we know, we understand that you are not able to solve the question. Sometimes you get confused which strategy to use while solving the question, which trick we need to apply, how to solve the question in less time, right? What type of questions are asked in the exam. So therefore, in this particular series, we are discussing the questions unit wise, topic wise, subunit wise, right? So please utilize this the sessions that we are conducting to the fullest, right? So at least try. Okay, so here I'm getting the answer option 2. Okay, option 3 also. So let me quickly brief you about what hybridoma technology is all about. See, monoclonal antibodies, when you talk about, we have monoclonal and polyclonal. Monoclonal antibodies, they always arise from single clone of cells, okay? That is the plasma cells. Whereas polyclonal, they arise from many clones of plasma cells. So plasma cells are short-lived antibody producing cells. Now, this hybridoma technology is popularly being used for synthesis of monoclonal antibodies. So here you can see all we need to do is that we need to inject the antigens into the model organism. So mouse, here it is shown, and this mouse will develop the antibodies, right? So basically you will be isolating the plasma cells, and these plasma cells are basically HGPRT positive. And another type of cells that you will be taking is myelonoma cells. Now these myelonoma cells, they are not HGPRT positive. Now when you are fusing these two, you are getting the hybrid cells. Now basically what is the characteristic of the myelonoma cells? That they are immortal, right? And here, what will happen when the hybrid cells are formed? So basically the fusion of these cells will happen in presence of polyethylene glycol. Now you want to check whether your hybrid cells, like finally what you get is the should be hybridomas. But maybe some of the cells they don't fuse. So you will be having a mixture of plasma cells, melanoma cells, and hybridomas. So for that we have to perform the selection so that we can separate the hybridomas from rest of the cells, plasma cells and melanoma cells which are unfused. So when you are growing in hat medium it is hypoxanthine, aminopterin and thymidine. Now basically aminopterin, it is the inhibitor of the DNA synthesis pathway. Okay, that is de novo DNA nucleotide synthesis pathway. You know very well that DNA synthesis is very important for the cell. If DNA synthesis is inhibited, cell will also not survive. Now, basically, if this de novo DNA nucleotide synthesis pathway is blocked, the cell will take up another alternative pathway for the DNA synthesis, and that is salvage pathway. And for the salvage pathway, the hypoxanthine and the thymidine, which is added in the medium, will be required along with the enzyme that is HGPRT. So here, if you see that once the fusion happens, your hybrid cells which are formed, they are HGPRT positive, right? Because your B cells were HGPRT positive. And what characteristic of melanoma cells we are using? That they are immortal, right? So what will happen? Unfused B cells, they undergo normal cell death, right? They will not survive for long period of time. Melanoma cells, they can survive, but in hat medium, they will not grow because aminopterin will block the de novo DNA synthesis pathway. So only hybrid cells will survive because they are both immortal as well as they are HGPRT positive. So this entire process was not required. Only the thing that we need to recall here was that what is the role of aminopterin here. And the role of aminopterin was to block the nucleic, like in the question, if you see, block the pathway for nucleotide synthesis. So this is the reason the answer here is option 2. So majority have answered it correctly. Priyanka, I hope the answer is clear why it is option 2. Moving over to another question. Now, sometimes, although like in the syllabus, only certain techniques are mentioned, right? But sometimes in CSIR net, some related questions based on some related techniques can also be asked, like ELISA spot, right? Basically, you need not to go in detail, but you need to be familiar with what this technique is all about. So either the complete question can be on that, or maybe the CSIR net in the exam, the question is framed in such a way that in one question they'll mention different immunological technique. Like one such question is shown over here. In the question, RIA is also given, ELISA is also given, ELISpot is also given, and Western blot is also given. So unless and until you are familiar with the principle of each and every technique, application of each and every technique, then only you can answer such questions. So this is the reason in the very beginning I told you that if you're preparing for this subunit, you have to study all the techniques given in this subunit. So the question is about that some statements are given, 4 statements are given about the features of immunoassay. So you have to go through each and every statement, all the 4 statements, and you have to find out the combination of the correct statements. So definitely to answer this question, you have to read and understand each statement thoroughly, and then you have to answer this question. So take your time and answer this question. So I think statement A, you know very well how to answer. What you need to recall here? The table that I showed you, right? In the table, the sensitivity of RIA was given, ELISA was given. See, these techniques, be it ELISA, be it RIA, they are sensitive and specific technique. But when it comes to comparison, okay, so which one is better? Which one is more sensitive? Every technique has its own advantage and limitation. It's not that all the techniques are perfect. There are certain, like every technique has its own advantage, has its own limitations, right? And this is the reason, because of some limitations are there, the scientists, they come up with the advancements in the conventional techniques, right? They modify the conventional techniques. Like in ELISA, you have many variations, right? You have direct ELISA, indirect ELISA, sandwich ELISA, competitive ELISA, right? Okay, Reshma, it's quite confusing. Yeah, I can totally understand Reshma, and this is the reason I have specifically selected this particular question for today's session, because I know I want to tell you the strategy of solving this question. So just try, okay? Just think about what could be the answer. Like, please note everybody, this guesswork you don't have to do during the exam. Right now, as we are practicing, and more you practice, more you learn. So that is why I always say that try. But in the exam, during the exam, if you are not sure about the answer, if you don't know which technique is about, like what is the principle of a particular technique or application, don't answer that question, okay? Because otherwise you will lose marks in negative marking. But as now we are practicing, so it's better that you try, so that like more you try, more you learn, and definitely if you answer it correctly, it will give you confidence, right? So just try once. Okay, so for this I'm getting option 4 and option 2. So that means if you are saying option 2 and option 4, so D is common. So that means confusion is between A and B. Okay. Confusing, sunshine. Okay. But what if I tell you this question is very easy? All you need to do is just recall, just remember one thing. That is the sensitivity of each and every technique. Okay, option 2 and 4, fine. So let's go, like we'll discuss statements given one by one, okay? So first of all, what is ELISA technique? Enzyme linked immunosorbent assay, right? And RIA is radioimmunoassay. Both are immunological technique. Immunological technique means where you used antibody to detect a particular antigen. And we use the antibodies which are specific for particular antigen. So obviously, when you are adding an antibody and the antigen is there, they will form a complex which is called as antigen-antibody complex. Now, if this complex is formed, we cannot see with our naked eyes, right? We need some signal generating system. We need which can indicate that okay, this complex is formed. So in ELISA technique, we use enzymes. The enzymes conjugated to antibody, and when you add a substrate, the enzyme converts the substrate into a colored product. So basically, by seeing the color, the product, colored product, you can find out that antigen antibody interaction is happening. And like an indirect ELISA, competitive in sandwich ELISA, the intensity of the color is directly proportional to the concentration of antigen. But in RIA, in radioimmunoassay, we use radioisotopes instead of enzymes. So the first statement is that radioimmunoassays are more sensitive than ELISA with chromogenic substrate. Chromogenic substrate means when the enzyme catalyzes the conversion of substrate into colored product. So colored product they'll form. So as I told you, if you see this table, if you compare RIA and ELISA, for sure the RIA is more sensitive, right? The RIA, it is more sensitive. And if you see the statement B, it is about ELISA with chromogenic substrates are more sensitive with chemiluminogenic substrate. Chemiluminogenic substrate means which emit light. Like the light is produced due to chemical reaction. Okay, like when enzyme basically catalyzing the conversion of substrate, so due to some chemical reaction, light is being produced, right? So that is chemiluminogenic substrate. Now, so here one state like one note I have mentioned that sensitivity of chemiluminescent based ELISA can be made to match that of RIA. So if you see statement A, for sure RIA is more sensitive than ELISA. So this is correct. But if you see statement B, that ELISA with chromogenic substrates are more sensitive, so this is wrong. ELISA with chemiluminogenic substrates are more sensitive than the chromogenic, ELISA with chromogenic substrates. So your statement A is correct, but B is incorrect. Then the third statement, the C statement, was about ELI spot, which measures the number of cells capable of secreting particular biomolecules using a substrate that gives soluble product with enzyme reaction. Now, here to answer this question, you should know what is this particular technique. So one thing, please note that this technique is majorly being used to determine the number of cells within a sample which secrete cytokines, right? Cytokines specifically. And here the technique is mentioned that what you need to do is you need to incubate, like you need to attach the antibodies, anti-cytokine antibodies, or I should say the antibodies specific for cytokines. You need to attach it to the multi-well plate. Now you will be adding the cells. Now the cells will not simply start releasing cytokines. You have to give some trigger, okay, so that the cells can start secreting the cytokines. So here you can see some cells will secrete, some will not secrete. Those cells which will secrete, you can see that the cytokines, they will now attach to the antibody, and then you can use the secondary antibody which is conjugated with the enzyme. So you will add another enzyme linked anti-cytokine antibody which is conjugated with enzyme, and you add a substrate, and this enzyme will catalyze the conversion of the substrate into colored product. So basically here, this is how the result will be viewed. It will not, like here you will the color will be basically, the result will be a blue black color precipitate will be formed. So it appears as a spot. It appears as a spot at the site of cytokine localization. So therefore statement C is incorrect because soluble product is not formed. It's a precipitate which is formed. And lastly, it is about Western blotting, that in Western blot analysis, the product of enzyme substrate reaction localizes at the site precisely where the antibody enzyme conjugates bind to its specific protein band. You should know that Western blotting is a technique which we majorly been used. It's a technique which we to use for protein analysis, right? For detection of a particular protein in your sample, protein of interest in your sample. So here again, I have shown the workflow of the Western blotting. All you need to do is you have to isolate the proteins from a cell. So many proteins are there in the cell. So you will be having a protein mixture. You will be separating the proteins on the basis of their molecular weight by performing SDS page, right? And so this technique I have covered in detail in the class. Okay. So here I will be telling you in short. So you will be performing SDS page, and finally you will be performing blotting. And blotting is where you are transferring the proteins from the gel to the membrane, which can be nitrocellulose or PVDF membrane. And then finally, once you transfer, when you perform the blotting, the last step is you will be adding enzyme linked antibodies. So if your antigen of interest is present, the antibody will bind to the antigen. And as your antibody is conjugated with enzyme, when you add a substrate, the enzyme will catalyze the conversion of the substrate into colored product. So basically, if your antigen of interest, your protein of interest is present, then yes, there will be a colored insoluble product that is deposited at the site of reaction. So basically, position of antibodies can be visualized. Basically, here you are using the reaction where you are reacting an antibody to an antigen, and that antibody is conjugated with an enzyme. When you add a substrate, it catalyzes the conversion of it in a colored product. So yes, antibody. So the question is about that your enzyme, product of enzyme substrate reaction localize at the site precisely where antibody enzyme conjugate bind to specific protein band. So yes, this statement is correct. So your A and D statement is correct, and therefore answer here is option 2. So those who have answered option 2, it is absolutely right. And I hope those who have answered option 4, now you understood why statement B is not correct. And Reshma, you have asked that, ma'am, instead of soluble, if they would have mentioned insoluble, then would statement C would have been correct. So statement C is Reshma. ELISA spot measures number of cells capable of secreting particular biomolecule. This is right. Using a substrate that gives insoluble product with enzyme reaction. So yes, then this statement would have been correct. So you can see, right, the CSIR net play with words. They are just replacing one word, right? And when you just change one word, the entire statement meaning gets changed, right? Okay. Thank you for confirming, Sunshine. So before moving ahead, I would like to tell you all about Watson Junoon Scholarship. So it's a scholarship program for graduates, postgraduates, and professionals. So those who have already enrolled, we really appreciate you all for that. But those who are not familiar with this scholarship or still have not associated, so it's a great opportunity for you all to associate with us. So still, we are taking the like you can register. Still, we are taking the admissions are we are taking the uh, graduates and postgraduates, and here are certain the advantage of this Watson Junoon scholarship is that you will be getting 1 year coaching for CSIR NET and GATE exam or QAT, IIT JAM and GATB exam. Also, you will be getting 12 months of access to all global and national workshops which are conducted by Biotechnica. You will be getting our programming internship with live project hands-on, and also you can attend the 4 certification courses of your choices. Even you will be getting access to the coaching experts and VIP pass to CRISPR conference. So for more details, you can contact on these toll-free numbers. So let's move over to another question. In radioimmunoassay for glucocorticoid hormone, radioactive glucocorticoid is added to RIA cocktail. When the amount of bound hormone was measured, no counts were observed. So following explanations were proposed. The radioactive hormone was insufficient. The radioactive tag to the hormones completely dissociated during storage. Antibodies not added to the cocktail, or the specific activity of the tritium was low. So here tritiated means that one of the isotope is replaced with the tritium. Okay. So you have to choose the correct option. So you can see, although this question is about radioimmunoassay technique, but the question asked is about why no counts were observed. So what do you think? What could be the reason? So if anybody is having the experience of wet lab, they can better relate to it. But you know, in CSIR net, we get the experimental based questions, right? So that is why we always say that if you're getting a chance to perform the experiments in wet lab, don't miss that, because definitely if you perform the experiments on your own, you can better relate with the theory, right? So theory and practical goes hands in hands when it comes to the techniques part. So what do you think? What is the answer over here? What is RIA? What is radioimmunoassay? It's a immunological technique, right? Where you use a particular antibody to detect a particular antigen, right? And basically, it's a very sensitive and specific technique. So this technique was developed by the two scientists, and they were developed. This technique was developed to detect the insulin concentration. So initially, this technique was developed to detect the insulin concentration in the blood. So you have to recall what RIA technique here, what RIA technique is all about. Okay, option 2, option 4. So option 2 is A, B, and C, and option 4 is B and C. So that means there is a confusion whether A is correct or not. So RIA we can use to detect hormones, right? Insulin, any particular antigen. Okay, Karen, you are saying option 4. Surendra, Suresh, you are saying option 4. Priyanka, you are saying option 1, and Anupam, you are saying option 2. Okay, Hemlata, you are also saying option 4. Okay, great. Try everyone. Please try everybody. Okay, Reshma, you are also saying option 4. Fine. So again, to find out the correct option, we have to thoroughly go through 4 statements. Okay, one by one. So first of all, let me just make you all recall about RIA technique. So again, this topic I have discussed in the class in detail. So this RIA technique, it was developed by Rosalind Yellow and Solomon Burson in the year 1960. And as I told you that this technique was developed to detect the insulin in the human blood. So it's a very sensitive technique. You can detect the quantity as low as a few picograms of your analyte. Okay. So it's a very sensitive technique, and you can measure the concentration of antigen using this technique. So it could be like you can study hormone levels in the blood, or any antigen, right? Like hepatitis B antigen, insulin, And definitely you have to use antibodies specific for that particular antigen. So this technique is based on combination of three principles. First is immune reaction between antigen and antibody. So basically here in this technique, what we do is we use some standard antigens which we make them radioactive. So whatever antigen you are looking for, the same antigens in the laboratory, you use them as a standards and you make them radioactive. So for radiolabeling, you can use iodine 125 or iodine 131, and then you will use specific antibodies for them. So antigen antibody complex will be formed. Now, once this complex is formed, what you're going to do is you will be adding the patient sample, that is the serum. So when you will be adding the patient's sample, the serum, so we don't know whether the serum is infected or it is containing the antigen or not, right? Like infected in terms of we are looking for some virus particle presence, okay? Whether a person is hepatitis B, suffering from hepatitis B or not, okay? So in that terms. So here we are looking for hormone, right? So patient sample is being added. So if the patient sample contains the hormone, what will happen? These antigens in the patient sample, they will compete with the radio labeled antigens. So the second principle is competitive displacement reaction or competitive binding. So more the antigen in the sample, more they will compete with the radio labeled ones and replace them. So radio-labeled antigens are also called as hot antigens, and the unlabeled ones, that is the one in the patient sample, are called as cold antigen. Now, once this displacement happens, after that you can measure the radioactivity of the free antigen or that of the complex, and you can separate the complex by using secondary antibody. So the third principle is that you measure the radio emission, radioactivity, and for that you can use the instruments like scintillation counter for measuring the radioactivity. And if you plot the standards, you can determine the concentration of antigen also. So basically here the first statement is that radioactive hormone was insufficient. So you need to understand that radioactive hormone is a standard hormone that we are taking, right? The standard antigen I told you, the antigen which you already have in the lab. Okay, see, you have, let's say, the hormone that you want to detect, the same hormone you will be using in the laboratory as a standard, and you will be making it radioactive. So if the radioactive hormone was insufficient, so you know radioactive hormone, if it is insufficient in quantity, even then you will get lower radioactivity, right? Some counts you will get, but it is not possible that you can will getting zero counts. No, even whatever radioactive hormones are there, even if you are measuring the radioactivity of the free antigen, so here antigen is hormone I'm talking about. So even if they are displaced, right, even if they are in the supernatant, then also if you measure it, you will get some radioactivity, right? Or if they are attached to the complex, then also we'll get some radioactivity, right? So no, not zero count, that is not possible. So A is incorrect. Then when you say that radioactivity tagged to the hormone has completely dissociated. So yes, this could be the possibility. This could be the most likely explanation for absence of counts. Because see, whenever you are performing these experiments, you have your radio labeling that you are doing, okay, or radioactive tracers that you are using, they need to be fresh, freshly prepared or ordered, okay. So radioactive tracers, basically the radioactivity degrade over time, right? Because radioisotopes undergo decay, right? So because, and even there's other factors like if the improper storage conditions are there, right, some chemical reaction happens that also results into the degradation of the radioactive tracers. So if the radioactive tag completely dissociated from the hormone molecule during storage, so definitely no radioactive signal will be there to measure and resulting into zero count. So yes, this could be the answer. Then you have statement C, antibodies not added to the cocktail. This mistake, like unless and until you're deliberately doing it, then it's the other thing. Because see, you have to understand in RIA, antibodies are the crucial components. If you are performing RIA, that means you need antibodies, right? You are not going to prepare this antigen antibody mixture then, right? Radioactive tracers, that is the antigen labeled with the radioisotope and the antibody. Your process starts with that, right? The RIA technique starts with that. Process starts with that, right? So If antibodies, which are crucial components in RIA process, were not added to cocktail, the binding between the hormone and antibody wouldn't occur. And yes, in that case also, there will be no counts. And last is the specific activity of the tritium was low. So even if the specific activity of tritium was low, you will get some few counts, not zero counts. So that means the most appropriate answer here, the statement here is statement B and C, not A, not D. So therefore, the answer here is option 4. So those who have answered option 2 and option 3 or 1, I hope it is clear why answer is option 4. Moving over to another question. Now, this question is about Western blotting. Now, if you read this question, basically, in the question, it is not mentioned it is Western blotting. But if you read the question, if you have studied the technique, you can easily make out that this question is about Western blotting. So very first thing is, if such questions are given to you, you have to read the question carefully and find out that this particular question is based on which technique. So here the question is that in a confirmatory test for HIV, one or more viral antigens are detected in the blood of patient and following other steps to be performed for the experiment. So as I always say, when it comes to technique, definitely you need to focus more on principle and application of the technique. Protocol is not needed, but at least you can go through the overview of the process. So here the overview is given to you and you have to find out the correct sequence of steps to be performed for the experiment. So go through the steps A to D and tell me what is the answer here. Okay, so for this, I'm getting very quick answers. Saujanya, Anupam, Sunshine, Surendra, Suresh, Archer, you are saying option 3. Fine. So if you go through the steps, you will able to understand that this particular question is based on which technique. And what you all need to do is just recall the overview of the process of that technique. Even I have discussed few minutes back about the Western blotting, right? I gave you in brief, I told you what all the steps are, right? So mainly when you talk about the Western blotting, it is based off 3 principles. First is electrophoresis. Second is blotting. And third is antigen antibody reaction. So all you need to do is first, as here we are testing for HIV, so you need to have a sample, right, which might be infected, right, which might contain this viral antigen. We have to confirm, right, whether it is present or not. So all you need to do is first you will be taking out the like you will be you need to have a sample and you will be loading your sample. So first you need to perform the electrophoresis, right? And once the electrophoresis is done, then you will be performing the blotting where you are transferring your antigen onto the membrane. And you can even block the membrane because you want your antibody when you will be adding to bind to the specific antigen and not to anywhere else on the membrane. So you can even block the membrane by adding BSA or milk casein, and then you can add the antibodies. So direct method or indirect method, you can use secondary antibodies which are conjugated with enzyme. So here we have used HRP. And finally, so horseradish peroxide, and finally you will be adding the like the chemiluminescent substrate, HRP substrate. So if you look at the steps, so first you will be separating the viral antigens, right? The next step is you will be transferring it to the membrane. Then you will be incubating with the antibodies specific for antigens. And fourth, detecting the bands by enzyme linked secondary antibody. So the sequence is C, A, B, D, which is given in option 3. So yes, majority of you have answered it correctly. So you can see in option 3 and 4, there is a slight difference. Okay, it is CABD, this is CBAD. That is B is the second step. So no, you don't incubate with antibodies first. You first transfer it to the membrane, right? The nitrocellulose paper. So it's time for another question, which is based on flow cytometry. So please go through this question and tell me what is the answer here. A mixed cell population was stained with two antibodies, one specific for cell surface antigen A and other specific for cell surface antigen B. So basically there is a cell having antigen A, another cell having antigen B, and you will be, so mixture of cell population is there, and for that you will be adding the antibodies, right? So one will be anti-A antibody for A antigen, another will be anti-B antibody for B antigen. The anti-A antibody was labeled with fluorescent. So this is labeled with fluorescent, and anti-B antibody was labeled with rhodamine. The cell population was analyzed for the presence of antigens by flow cytometry. What is the correct outcome for this cell population? Some data is given to you. So you have to analyze it and tell, like, basically these are the outcomes. So you have to tell which is the correct outcome for the cell population if such kind of analysis of the cells of the antigens is done. Sunshine, just try to check your settings because it is looking fine to me when I'm checking it. Okay, option C, some are saying option A. So if you have attended the regular classes, I told you one trick, right? I told you to remember one particular diagram, right? When we were discussing about dot plot analysis, I told you that. So you just have to recall it here and you have to answer it. Okay, so majority says option C here. Fine, great. Yes, absolutely right, Komal. Double positive and double negative. Just recall which quadrants double positive and double negative are present. So I'm sure as you are answering the question, I'm sure you know what is flow cytometry, right? So flow cytometry, as you all know, it's a very powerful tool used by immunologists to study multiple parameters on the cell. What are these multiple parameters? You can study the cell, you can identify a particular cell based on the particular marker or antigen on its cell surface. Even you can identify the cell based on the size and also complexity or granularity. So you can study parameters like complexity, the size of the cell, and using flow cytometry, it's a quantitative technique. So you can count the number of a particular cell in the mixture, okay, in the mixture of cell population. So we can characterize a particular cell, right, by looking for a particular antigen on its cell surface or maybe intracellularly, and for that we'll be using the antibody, because flow cytometry is again immunotechnique where we detect a particular antigen unique to a particular cell by using antibody specific for it. But as I told you during this session, that antigen antibody we cannot see with our naked eyes, the complex which is formed, we need some signal generating system. So in flow cytometry, the principle which is followed, which is used, or I should say the flow cytometry is based on principle of immunofluorescence. So you use fluorescent dyes here. So flow cytometry, it's an analytical technique. You can quantify the frequency of cells and which are binded to a particular fluorescent antibody. And even we have the advancement in the conventional flow cytometry, using which we can even sort the cells in different containers. So the advancement in the conventional flow cytometry technique is FACS, that is fluorescence activated cell sorting. So as you can see here, we have, let's say, the cells, right? So some cells might be having only A antigen, right? So, and some cells might be having only B antigen. So as you can see here, some cells are having no antigen. Some cells are having only B antigen. Some are having only A antigen, and some are having both A and B antigen, right? So I can write it like this, like those cells which are neither having A antigen and B antigen, I can label it like this, right? So please note here, this minus is not a charge. Minus means absence and plus means presence. So so we are considering four type of cells. First, cells which does not have a A antigen and not B and also not B antigen. Another cells which are having A antigen but not B antigen. Third type of cells having not having A antigen but B antigen is there. And fourth type of cells which are having both A antigen also and B antigen also. So basically, you will be introducing the cell you are mixing like you have a cell population. You will be adding it into the flow chamber, the flow cell of the flow cytometer, and inside the arrangement is like there is a sheath fluid, and the arrangement is that there's a vibrating nozzle, and the arrangement is that the cells align in a single file, and they pass, like they align in a single file and they pass one at a time through this nozzle. And as they are passing, we are passing the laser beam. So basically here, these antibodies which we are using, they are conjugated with the fluorescent dyes. So as mentioned in the question, for NTA, like we are using a like fluorescent and rhodamine. These are the two dyes we are using in this experiment. So as the laser beam passes through it, fluorescent dye absorbs the light and emit light of longer wavelength, which are detected by the detectors. And even we have the deflection plate. So as the cells they are passing, so they have a particular charge based on what fluorescent molecule they are being labeled with. And based on that, a charge develops on a cell and they can be sorted in different tubes. So this is the dot plot which is being shown, and I shared a trick in the class which I would like to share it again here. So I told you that what you all need to do is just draw the square divided into four quadrants. You have four quadrants. You have upper right, you have lower right, you have lower left, and you have lower upper left. So always remember, like if you are plotting NTA, okay, if you are plotting NTA fluorescence here, that is NTA antibody labeled with the difluorescent, right? So fluorescent emit green fluorescence. So basically, just consider it to be a starting point. So obviously, like we do in a number scale, right? Like in a number scale, if this is the zero, any value more than zero is positive and any value less than zero is negative, right? So same thing you have to keep here in mind, that if this is the initial point, so here we are on x-axis as we are looking for NTA, and like we are looking for green fluorescence, which is indicating the presence of A antigen. So this indicates presence of A, and this quadrant will indicate absence of A. Similarly, on y-axis, we can plot NTB antibody, and the NTB antibody is labeled with the rhodamine, which emit red fluorescence. So again here, we have here you can say this quadrant will indicate absence of B, but this quadrant indicate presence of B. So if you write the combination, so this quadrant will be your double negative always. This quadrant will always be double negative. This quadrant will be where A is present indicating but B is absent. Then if you see this quadrant, here B is present but your A is absent, right? As shown over here. And the last quadrant, which is double positive, where you have, which indicates both presence of A and presence of B. So just this trick, if you remember, you can easily answer this question. So although all these things are not mentioned in the question, that four cells you have to consider, but this is the way, based on the result, the outcome which is given, based on that you have to recall this information, and you have to remember that this is the diagram you all have to recall. Now once we have drawn this diagram, all you need to do is just see that in the outcomes given in the question, which one is resembling to our outcome. So here you can see this is double positive, this one is double negative, and you can see this is indicating the presence of A antigen, and this is indicating presence of B antigen, right? So here, if you look at the A, B, C, and D outcomes, you can see this is the C outcome, which matches with our result. So therefore, answer here is option C. So it's easy, right? If you remember about all these points which I'm telling you, the tricks I'm telling you, you will never, like, you can all answer the questions which comes in the exam. So keep the points which I told you during this session, the tricks, the strategies of solving the questions. And I hope you find this session helpful. So please let me know in the chat. Did you enjoy today's session? Yes, Reshma, just like Ramachandran plot, right? So for Ramachandran plot also we use this trick. You can use this trick, okay? So yes, do you find it useful? Do you like today's session? Okay, that's great. Thank you for your response. So we'll see you in another session where I will be discussing some more questions on histochemical and immunotechniques. Thank you for attending this session. And just remember that to accomplish great things, we must not only act, but also dream, and not only plan, but also believe. So believe in yourself. Thank you, everyone.